We'll bring on our next presenter, which is actually quite good timing because he's going to be talking about pre-recorded data. So, and that's Neil Ward from Massey University. Massey is, a bit of a hotspot of very innovative users of LT. So, yeah, really look forward to Neil's talk. Hopefully Neil will be coming on soon. There we go.
Right, I'm just clicking buttons here. Now we're right.
Oh, welcome, Neil. So I'll hand it over to you, Neil. Cheers.
Okay, uploading me. I'm going to share my screen first, don't I?
Yeah, could help. Cheers.
Right, now we're in business. Okay, well, good morning. Yes, obviously I work at Massey University in the veterinary school, and my role is in the physiology laboratory support and a whole bunch of other teaching and research activities. But my presentation today is just to run through what was our first draft of creating a virtual lesson entitled Virtual Visceral Smooth Muscle. And it was an attempt to integrate the learning of the structure or the microstructure, the function, and some of the basic pharmacology or the autonomic pharmacology of smooth muscle. The lesson starts with an obvious introduction. Clearly the benefits and drivers for this sort of approach were in keeping with the rest of this meeting. Everyone's been dealing with the COVID issue, so we had to switch to be able to try and deliver things online. Massey also has a multi-campus presence. We have a campus in Auckland and one in Palmerston North, which deliver a physiology paper or physiology courses are the same. So we were able to deliver exactly the same practical course experience at both campuses. And the other bonus, the benefit that's come from this, of course, is that we're replacing the need to use live animals. So we started off with a basic structural overview. So pretty picture of rabbits and some cartoons and some histology. So to give an orientation on what we're dealing with here. And then we move into the histology, the microstructure lessons, which was taken from a series of lessons on smooth muscle, skeletal muscle, and cardiac muscle, which was formed the basis of histology teaching. So the lesson works through obviously basic information, cartoons, and we utilize our virtual microscope system, which is an online microscope called PathoBin, and that's via a Melbourne donor. We get it for free. So the students are able to go to their slide that's linked and take a screen snap, and we'll see later how we asked them to annotate things. There's obviously the ubiquitous drag and drop questions that exist in LT, which are very good. So that forms the basis of learning. And here's the first upload screen snap. So we've asked the students here to take a screen snap and upload the snap and identify the myenteric plexus. Whoops. What's going on here? So they've got to identify that, and they can, of course, give it a name or a label. And a check answer, usual story as well, thanks to the tool sets there. Another example is looking at the villi structure. So we have another case here where we invite them to upload another screen snap from another slide and clearly label a villus. And likewise, they can label brush border. So a little bit of active learning going on in these lessons, which is always a good methodology. Further drag and drop questions just to reinforce their knowledge about other components of the mucosa, and a few other options that they work through as well. Then we move across to the neural and endocrine control of a piece of, well, of GI tract smooth muscle in general. So here we have an overview of information on the basic electrical rhythm, which drives the rhythmic contractions, and the concept of reaching a threshold and getting action potentials which cause contractions. A little bit of a read on the autonomic control, and some more overview here on the aspects of endocrine control and autonomic control. One of the lecturers decided to put this in just to start to look at the receptor level, the membrane level, through to propagating actin and myosin response. Again, that's sort of a review of what they would get in lectures. It's just a sort of a reminder, really. Then we look at the virtual experiment. So this is the setup that we used to use for the hands-on experiment. And so it's just taken from those hands-on practicals. So they get a heads up on that. We obviously need to describe what we're trying to do. We've obviously got to keep the tissue oxygenated at constant temperature, and it's in a physiological solution to ensure that the tissue is able to thrive. And in our lab, we had these instruction panels to help the students prepare their tissue and put it in a bath, so they would work through that. And then this is a new bit we've describing what we're doing with a virtual experiment. So obviously it's just saying, well, we've pre-recorded things and set it all up. And then we move across to our virtual experiment. So the first thing is to observe normal rhythmic contractions. And with the tools, of course, we can get some measurements of force of contraction for comparative purposes, and frequency changes if necessary. And of course, the students are required to make a response and answer to what they see. The video is just simply exporting the video from chart file, and that's scrolling at real time. We could have accelerated that, but we didn't. It's a slow event, so it's probably realistic to see it running in slow time. Textbook sort of physiology working through here with an injection of acetylcholine. Start to play around with the autonomic control and the neurotransmitters that are at work here. And again, they can observe that there are frequency changes and so on. We did a sort of a dose response curve to show that more is better. So a bit more acetylcholine produced a slightly bigger response. And then in line with looking at the autonomic system, we could see that adrenaline inhibited the contractions of smooth muscle. And again, they can make some measurements and make an observation. We're always trying to make them unpack the physiology and write it down, and so on. So we then move through bigger doses of adrenaline, atropine injection to show competitive blocking action. We did a dose response curve on the atropine dose. Ezerin was an interesting one. That's just simply to show the abolition of the cholinesterase and a slow but progressive increase in contractility due to the accumulation of endogenous acetylcholine. So that's a classic sort of message there as well. And then we let them play with potassium and calcium injections or infusions to try and understand changing membrane, resting membrane potentials, and how the effect of that would influence the contractility. So thanks to the team, Dr. Preet Singh and Anthony Jacob, the three of us were involved in creating this particular one, and we've done some other virtual experiments on cardiovascular physiology, ECG work, and on blood pressure control. So that's a little run through for our first draft. Thank you very much. Any questions?
Awesome, cheers Neil. Yeah, cheers for burrowing down and showing LT in much more detail, particularly the data panel aspect, because there's probably quite a few people that may not have seen that, and there's probably also quite a few people that want to geek out with you on the pharmacology aspects of that as well. We do have one question from Ricky. So she says, thanks for sharing, very interesting. I remember this lab from 1993. How were lectures delivered under lockdown?
During the lockdown, all labs were
lectures.
And lectures were done by Zoom or just with documentation that students could download. But yeah, it was a mad panic for everyone to get up to speed with Zoom and try and do some sort of synchronous experience for the students. So yeah, Zoom was the savior, and right through the vet teaching, our staff, not only in physiology, but our staff got into using LT in a big way and got quite creative. So that was a bit of a saviour as well for sort of practical tuition type work.
Awesome, cheers. So yeah, bang on time. Helps if I unmute myself.
Yeah, yep.
Thanks, Amy. It's all right. Yeah, speaking of Zoom, thinking I'd know how to unmute myself by now. Yeah, so thanks, Neil.